All right, let's go ahead and get started. So before I go into lecture mode here, we are going to do part 2 of the content check together in class, and we'll do that at the end of class. But I want to give you about 15 minutes to do it, so if you notice I'm talking past like 11:25, flag me down and say, hey, it's time for content check. I don't know how far in the lecture we'll get. Any questions? All right. Well, we're going to continue with the last bit of lecture from Wednesday. So Wednesday we talked about carbohydrates, right? But we did not quite get to lipids. And so we're going to try to get through the last 3 of our major biomolecules today. We'll see how far we get. But starting with lipids here. So lipids, right? These are going to be molecules that are mainly carbon and hydrogen linked together via covalent bonds. Right? A large portion of them is just carbon and hydrogen. That makes a large portion of these molecules nonpolar. Right? We can see these nonpolar regions in this like purple-pink color in this image here. However, on most lipids, they're also going to have an area where oxygen is also covalently bonded. Right, now oxygen is going to make that part of the molecule polar. Right? And if we look at this part down here, it's kind of like this side part in the way that this image is oriented, that is polar. Right? So the area with the oxygen will be polar, and then they'll have a fairly large non-polar region. This means because a lot of their region is non-polar, they tend to be hydrophobic, or water-fearing. Right? When we're thinking about lipids, we're thinking like our oils, our fats, and our steroids. Right, so covalent bonds, they have a polar and non-polar region. They tend to be water insoluble. There are different types of lipids that we will look at. So lipids, they're going to vary in what they look like and how they function. There are 3 types that we are going to go over, the 3 major types, and that's fats, phospholipids, and steroids. Now lipids tend to be different from a lot of the other biological molecules in the fact that they are not large molecules and they are not built from monomers. Right? So on Wednesday, we talked about carbohydrates. They're built from monosaccharide monomers, right? We string those monomers together to get the polymers. Lipids are different in this function, right? They are not built from those monomers. So we might have fats, also known as triglycerides, right? Steroids, like cholesterol, important in hormones and cell membranes. Phospholipids, they play a major role in the plasma membrane of a cell. We're going to start with that top one there. We're going to start by talking about fats. So how are fats different from the other lipids? Well, fats, they're also known as triglycerides, right, are formed by bonding a glycerol molecule to 3 fatty acids. All right, so we have our glycerol molecule. Okay. It's going to bind to these fatty acid molecules via dehydration reactions, so we're using some water, to create a triglyceride. Okay. Now, triglyceride, tri meaning 3, I think 3 fatty acids. Okay. So we have our 3 fatty acids joined to our glycerol via dehydration. They can always break apart via hydrolysis. And the big part that they play is that they store energy. Right? They can store energy that then the cell can then go back and break apart. And when they break it apart, they're releasing energy and they can use that energy for cellular functions. So when we're looking at molecules like this, I would want you to know the components of the molecule, but I would not necessarily want you to draw out the molecule like they have drawn out down here. Rather, I'd want you to know that it's a glycerol with 3 fatty acids, rather than drawing the entire chemical structure out. Now, fatty acids can differ, right? So we can have saturated and unsaturated fatty acids, and that's going to be based on the number of hydrogens bonded to that carbon-hydrogen chain. So before you start with saturated fatty acids, right? A saturated fatty acid is going to have the maximum number of hydrogens that could possibly bond to that carbon backbone. And so if we look at our triglyceride here, 2 of these, the first 2, are unsaturated fatty acids, right? They have the maximum number of hydrogens that could possibly be bound to the carbon. These types of fats tend to be like animal fats, like butter or mallow. It takes a lot of energy to melt them, which is often why we might find them solid at room temperature. Right? If you put butter out on the table, it's not going to immediately melt and create a huge mess. Right? It might melt a little bit depending on the temperature, right? But it's going to pretty much stay solid. All right, let's compare that to unsaturated fatty acids. Unsaturated fatty acids do not have the maximum number of hydrogens that they can hold because carbon is making a double bond somewhere within the fatty acid. So if we look again at our triglyceride here, this third one, this is an unsaturated fatty acid because in this case, carbon has made a double bond with carbon, which means these 2 carbons, the way that they're bonded together, can only hold 1 hydrogen each instead of a usual 2 each. Right? So there was a possibility for it to be saturated, but because of that double bond, it's unsaturated. These tend to be a lot of oils, like canola oil, olive oil, safflower oil, right? Oils that are not solid at room temperature. The biggest difference there, saturated, unsaturated, the number of hydrogen bonds. If it has a double bond somewhere, right, it's not going to be holding the maximum number of hydrogens that it can be holding, which will make it unsaturated. All right, we're going to do a group question next here. And in this group question, I have given you a triglyceride. I want you to figure out how might they arrange themselves together if I were to put 8 of these in water. Now, this is a lot to draw, so you can draw it shorthand, right, at this polar head and this non-polar tail. So give it some thought. See if you can't figure out how they might arrange. Work with one another. Talk it out.
It looked like that. The first two would be like great. And that one on the balance on.
This is a trickier one. I'm asking you to apply concepts from previous lectures. As a hint, think about the polarity. How might this molecules orient themselves in terms of polarity? If I give you this molecule and said green fatty acid connected to glycerol, what is it? You tell me fat, or if you told me triglyceride. This question ended up being a little bit trickier. I'm not going to give you more time just because I want to make sure we get through some content so we can do that content check. But does anybody have an idea of how these 8 molecules might arrange themselves? The nonpolar. Yeah, that's exactly it, right? If we think about polarity here, the nonpolar bits, they are water, or they're hydrophobic, they're water-fearing. They don't want to interact with water. But the top part here, right, they have a polarity to them, which means they're water-loving. So what's end up going to happen is that they're going to form these circles where all the tails are inward. So nonpolar is next to nonpolar. They want to stay as far away from the water as they possibly can. Where the regions that are actually water-loving and don't mind interacting with water will be outwards. Right? Does that make sense? We saw something similar when we talked about mycelials on the last Friday. Right? This is drawing a lot of different concepts together. This would be a great conceptual question for an exam because it's asking you to put many different things that you've learned together. All right. So that's fats. Right? And that question leads really great into our next type of lipid, which is a phospholipid. Now, a phospholipid is really similar to a fat in its structure, but instead of 3 fatty acids, we only have 2. They're still connected to a glycerol molecule. Right? So a glycerol with 2 of these fatty acid tails instead of 3. And they have an addition of a phosphate group on the other side of the glycerol. Right? So again, they're going to have a polar and a non-polar region. Right? You know, carbon and hydrogen, those covalent bonds, those electronegativities are really similar. Right? So they're going to be non-polar regions. Versus up here, we have lots of oxygens. Right? They are very highly electronegative, which means they're going to have polarity on this part of our molecule. We tend to call it the hydrophilic head and the hydrophobic tail. Right? Now, just like we saw for that triglyceride, that group question, those hydrophobic tails are going to want to orient themselves towards each other. Right? Kind of make a barrier so it doesn't have to touch water, essentially. Because it's hydrophobic, it doesn't want to interact with water. And we see this most commonly in our cell membranes, right? In our phospholipid bilayers. So when we look at a cell, they're surrounded by a membrane made up of these phospholipids. And again, the tails, they're pointed inwards. because the nonpolar regions want to be next to the other nonpolar regions. Versus the polar hydrophilic head, right, they don't mind interacting with water. They can interact with the extracellular or intracellular fluid that is typically largely water. Right? So again, polar and nonpolar playing a role here. Our last type of lipid is a sterol or steroid. These look very different. Right? These are not similar to fats. They are not similar to phospholipids. However, their arrangement is going to be in 4 rings of carbon. Right? So the lipids in this case are 4 rings of carbon. And depending on what is attached to those rings will give it its unique property. Right? It might make it a cholesterol, or it might make it a hormone. Cholesterol is a really common one. Right? You see cholesterol up here. Again, has that 4-ring base. And then this extra molecule that's added on up top here is what makes it cholesterol. A lot of animal cell membranes will have these embedded within their membranes, and they function a lot for like cell-to-cell signaling, so we can communicate with other cells. But it's also a starting material for many other types of steroids. Right? Steroids like hormones, like sex hormones, right? We have testosterone and we have estradiol. These are also steroids as well. The key here is that they're in 4 rings. You might be more familiar with steroids in the form of like athletics, right? And they're doping, right? Anabolic steroids is what you would be referring to in this case. They tend to be a variant of testosterone, and they can cause muscle mass to build up, which is why athletes might want to abuse them to be more muscular, right? But these anabolic steroids aren't all bad, right? There are some diseases like anemia or diseases that destroy body tissue where these individuals will take this anabolic steroid to build up their muscle mass just to be at a normal level of muscle mass. Right? So there are good things with these steroids. But if you abuse steroids, you often get really severe side effects, like violent mood swings, often known as roid rage, depression, liver damage, cancer, high cholesterol, and blood pressure. All right? So this is probably the version of steroids you commonly hear about in everyday language. Right? They can be good, though. They can be good, especially for individuals that have certain types of diseases. All right, we went through 3 types of our lipids. I want you to practice naming them. Again, this is a type of question that'd be really good for me to put on an exam. Just give you a structure and say, based on the structure, can you tell me what molecule this is? So I want you to label each type of lipid, and then if it has fatty acids, tell me if those fatty acids are saturated or unsaturated. So a bit of a review. a really good review here. What is my first molecule? What would I call that? Phospholipid. Yes, I could hear the S's very clearly in those whispers. It's a phospholipid. Yeah. Why is the reason why did we call it a phospholipid? What led us to that conclusion? Its shape, yeah. It has two fatty acids. But it also has this phosphate group on top. All right, what is the middle one? Steroid. This one should be an easy one. It looks very different from the others, which leaves our last one as our fat, or as our triglyceride. All right, let's go back to the phospholipid. Is the one on your right hand or your left hand side saturated? I think I heard left. Yeah, it's saturated. It has all the hydrogens it possibly could have. There are no double bonds. All right, which means the other one, because it has that double bond here, right, would be unsaturated. It has the potential that there could have been more hydrogens attached, but it made a double bond, which means we no longer can have any more hydrogens attached. All right, these triglycerides. What about these first two? What would we call these first two? Saturated, yep, again, because they have all the hydrogens they possibly can, which leaves that other one, again, double bond being the clue here, as our unsaturated. Yeah? How do I know that's a phosphate group?
Yeah.
Yeah. Well, part of it's just knowing the molecule, but the other part of it is I see a P in the middle, and that's shorthand for phosphorus. So that gives me a clue. That's fine, yeah. I mean, I expect you to take away the biggest clue from this one is the 2 fatty acid tails versus the 3. Any questions about lipids? We're going to do another question. And I'm giving you 5 minutes here. You might end up being done sooner. So, oil and water do not mix. We know this. Right? But mayonnaise actually contains both of these products, and mayonnaise does not separate out into water and oil. Right? This is because it has an emulsifier in it. Right? An emulsifier like this lectin is a molecule naturally found in egg yolks. So I want you to tell me, how does lectin promote the mixing of water and lipid molecules? Right? So you'll have to probably Google how does an emulsifier work. All right, so emulsifiers, how do they work? How are they going to promote the mixing of oil and water together? Who came up with an answer for this one? Yeah? Yeah, essentially the ones that don't like each other. Right? Like think about hydrophobic molecules. With hydrophobic molecules, they like each other, they're going to stick together. Right? And it's going to keep them separate from the hydrophilic parts of the solution. I have this a little image up on screen here that maybe helps understanding a little bit more. So if we have a globule of oil, oil is hydrophobic. It does not dissolve in water. This lectin molecule, the emulsifier, will essentially surround that globule of oil, right? So the hydrophobic ends are surrounding the hydrophobic oil. The polar ends are interacting with the water itself. Very similar to how we talked about like the mycelials in terms of dish soap and that grease fighting power that it has, except this time, instead of using it to wash away the oils, we're using it to mix the oils in with the water. We're not washing it out. Any questions on this one? All right. We're going to move on to our next biomolecule. So we're going to switch gears a little bit. We've done carbohydrates. We've done lipids. Now we're going to talk about proteins. Right? Proteins are such a hugely important type of molecule. Right? We find proteins everywhere. They function in a whole variety of roles. Right? They can be enzymes, which help speed up chemical reactions. They can be part of antibodies. They can help movement within the cell, structure, signaling, transport. Right? There's so many different roles that proteins can carry out. Right? Likely, if you were just to randomly sample a molecule in your body, it's going to be highly likely that you'll find a protein. Right? So many different functions. I don't expect you to memorize all these specific functions. As we go throughout the semester, right, we'll talk more in depth about enzymes, and we'll talk more in depth about movement. and how proteins are involved. Right, so vastly important. They have so many different functions. But what's kind of interesting and unique about proteins is that they're only made up of 20 monomers, 20 different monomers. So proteins are made up of amino acids. There are only 20 amino acids, right? These amino acids will have different functions to them, right? Some are going to be nonpolar, some are going to be polar. Some have a negative charge, some have a positive charge, right? They all have these different functions. And it's the structure that really is going to dictate that function. You're going to hear me say this a lot. Structure equals function. Right? So all the molecules, millions of types of proteins out there, they all have this basic component of just 20 different amino acids strung together in different ways to make different molecules. If we look at that monomer, which is the amino acid, if we look at the amino acid structure, it has a very specific structure to it, in that it's going to have a carbon in the middle, it's going to have an amino group on one side, which is this N connected to our 3 hydrogens, and it's going to have a carboxyl group on the other side, which is carbon and 2 oxygens. Right? So it's always going to have an amino group, always going to have a carboxyl group, it's always going to have this extra hydrogen here, and then this R, right, again means the molecule continues this way, will give it its specific amino acid distinction. So whether it's valine or proline, right, it's going to distinguish it from the other amino acids. And again, there's only 20 different ways that this can vary. So again, I'm not going to ask you to draw this structure, but I might ask you what makes up an amino acid structure. And you might want to say carbon in the middle, an amino group, our carboxyl group, and hydrogen, and a side chain or an R chain. Right? That is the underlying structure for all 20 of these molecules. Now it's when we start linking these amino acids together that we start to get proteins, right? So polypeptides is what we call the polymers for amino acids, for proteins. Right? Polypeptides are a chain of monomers, the monomers being amino acids in this case, joined by covalent bonds. Right? And just like we saw for carbohydrates, there's a very special name for when a sugar is bonded to another molecule. That's a glycosidic covalent bond. Right? This one also has a special name, which we would call a peptide bond. Right? Bonds holding amino acids together. So we start to build these chains of different amino acids. And they're built in the same way we've seen for lipids, same way we've seen for carbohydrates. These dehydration reactions, right? We're taking the OH from the carboxyl group. We're taking a hydrogen from the other side here, taking away water. We get a covalent bond, which we specifically call a peptide bond. Now, proteins have very interesting structures. Remember, they're only made up of 20 different molecules strung together. And it's really how those molecules interact that give it a specific structure that leads to its very specific function. So a protein, we would define a protein as one or more polypeptides, which are the polymers, that have folded or twisted, combination of both, one or the other, right? It's folded in a way that gives it a three-dimensional shape. Right, so we're not just talking about beads on a string here. It's going to fold in all these complex ways. It's going to get a shape, and that shape is going to determine how it functions. Is it an enzyme? Does it act in signaling? Does it help in movement? Oftentimes these proteins, right, they have these polypeptides, which are the polymers again, right? They'll often have other types of molecules attached, like carbohydrates or lipids, that also help determine some of those unique functions. All right, so we have our monomer, our amino acid. We have our polymer, our polypeptide. We put one polymer or more together. They fold, they create a three-dimensional shape. Now we get a protein. All right, there's a lot of different levels to how this shape might take form, right? There's a lot of folding, there's a lot of interacting going on between these amino acids. The first level of the protein structure we would call the primary structure, and that simply is the order of amino acids. Think that beads on a string molecule, right? This is our primary structure here. We have amino acid bonded to an amino acid, bonded to an amino acid. Just a string of amino acids. But we know it doesn't stop there, right? It's going to continue interactions between these amino acids beyond just a string of them. Right, this is where we're going to get our secondary structure. So now we're beginning our folding and our twisting. We're starting to get our three-dimensional shape. Right? Secondary structure now, we might see things like alpha helices, like in pink here. We might see beta pleated sheets. Right? They're starting to get a three-dimensional structure. Right? And this is all due to hydrogen bonding. Remember when we talked about hydrogen bonding, it's between a polar molecule and a polar molecule, but sometimes it can happen within really large molecules. This is an example of it happening within one molecule that's very large. Right? We have these amino acids, beads on a string, right? These beads, they either are polar or nonpolar. They're either charged and have a positive charge or a negative charge. Those charges and partial charges all start to interact with each other. So they can have some hydrogen bonding within one molecule to create these 3D structures. Okay. So again, these alpha helices, we have our hydrogen bonds creating these helices. These beta pleated sheets, we have the hydrogen bonds creating these plated sheets. We can also have other structures that are just generally coined randomly coiled regions. Right? But these are the two most prominent that we see. All right, so we took our beads on a string for our primary structure. We have started to fold it together in our secondary structure. Right? The next line of structure would be our tertiary structure. Right? We're going to fold this line of amino acids even more. Right? So we have our beta pleated sheets. We have our alpha helices. Now we're going to have interactions between those beta pleated sheets and those helices. They're going to start interacting with each other within that same molecule. So again, even more 3D structure in play. This is really where those charges play, again, a huge role. Right? We have positive charges, we have negative charges. Opposites attract. Even if they're in the same molecule, right, those opposite charges are still going to attract, still going to create more complex 3D structures. Right? So far we've been talking about a single polypeptide chain. We've taken it from amino acids, beads on a string, to folding it into things like beta sheets and alpha helices. Those alpha helices and beta sheets are now interacting with each other in this tertiary structure. And this is the final level of structure for a single polypeptide chain, right? For a single polymer. But this is not the last level of structure for a protein, right? We get even more complicated. Our last type of structure is quaternary structure. This is where we have our polymers that have been folded into a 3D shape. And now these polymers are interacting with other polymers to create one large protein. Right? So we have those individual polypeptide chains that are folded in these complex ways. We would call them a protein subunit. Right? these polypeptide chains interact with other polypeptide chains to make a molecule, to make our protein. A good example of this is hemoglobin. It has 4 different subunits, which means it has 4 different polymers that are bound together into one protein. Hemoglobin is a good example of a protein that is multimeric, which means it doesn't just consist of the same polymer bound to the same polymer, but rather it's one polymer bound to a different type of polymer. And in this case, it has 4 subunits. So we have an alpha subunit, right? Alpha 1, alpha 2. These are the same polymers. We have beta subunits, beta 1, beta 2. These are the same polymers. So there's 2 different types, but we have 2 copies of each. Together, they make hemoglobin, which we can find in our blood. All right, so it just gets more and more and more complex. Think about we're building upon each other. I'm trying to think of an analogy off the top of my head. Oh, I like to play Legos, so you can think Lego. We have one Lego brick, we stack it on top of each other, right? There's only so many Lego bricks that we can have that are created. That would be the primary structure. Now, maybe we've created several structures, and we want to link them together. Maybe that's our secondary structure, right? We can take several of these linked structures and interplay them with one another, again, making it more complex for our tertiary structure. And then finally, maybe we have like 3 different Lego sets and we're putting them all into one big build, all right? And that might be quaternary structure. It's the best analogy I can come up on the top of my head here. All right, let's do this group question, and then I think we're going to go on to the content check after this. But in proteins, what are the monomers? What are the polymers? Right? Quick review. And then imagine that a protein's primary structure was altered. Would this affect its tertiary structure? And why? Or why not? tertiary. Yeah, yep, rather than there being ions trapped in it. Yep. All right, if I were to ask you, what is the monomer that makes up a protein? What would you tell me? Amino acids, yes. And those amino acids, when we make a polymer, what do we call them? Polypeptide, yes. So if we think about the way that these polypeptides fold and create these three structures, if a protein's primary structure, right, so the amino acids on a string, were to be altered, would it affect its tertiary structure or its 3D shape? Yeah, why do we say yes? Why is it a little bit more tricky? Yeah, if I use the wrong Lego piece, it would mess up the whole set. So an example of this, and something that you'll start to see in lab next week, is sickle cell disease. So sickle cell disease, in a normal red blood cell, right, we have the protein, I think this is glutamine, GLU, right? But there's been a mutation which causes just this one singular amino acid to be replaced by a different amino acid, valine. That one single replacement affects the way that those R groups, which typically have the charges or the partial charges, interact with other amino acids, how they fold, and now we get a sickle red blood cell, right? Completely different shape, just because we changed one amino acid. All right. Before we move on here,